The following video details a procedure for using the CBRIC standard to assemble DNA parts using CRISPR-associated protein Cpf1.
This method can further the development of DNA assembly in the field of synthetic biology, such as by facilitating the construction of complicated genetic circuits. The main advantage of this method is that it is sequence independent and allows for iterative assembly of DNA constructs. We first had the idea for this method when we noticed that Cpf1 was guided by single cRNA and cat-target DNA in a stacked way, therefore generating sticky ends. Demonstrating the procedure will be Shi-Yuan, a graduate student from my laboratory.
To prepare the crRNA templates, resuspend the individual oligonucleotides in RNase-free water to a concentration of 10 micromolar. To a 0.2 milliliter PCR tube, add top strand oligonucleotide, bottom strand oligonucleotide, and 10x annealing buffer as detailed in the accompanying document. Ensure that the total volume is 50 microliters. This table shows 6 different bottom strand oligonucleotides, each of which should be individually paired with the top strand T7F. The lowercase letters represent the sequence to be transcribed into the guide sequence of crRNA. Place the PCR tube into a thermocycler. Run the annealing program with an initial denaturation at 95 degrees Celsius for 5 minutes and then a cool down from 90.